Hello students, welcome to Biosastra Talks. I hope all of you are doing well and good luck for the NEET exam as we are coming nearby. So that's why I thought to give you some FAQs or PYQs which will commonly present or come from the important chapters. Okay, so today we'll learn about what were the frequently asked questions from the genetics chapter. Why? Because we know that genetics is the most high weightage one from the botany, okay, for the NEET exam. So let's get into the FAQs that came from the genetics. Okay, so what was the first question was this question was given in the NEET 2023 paper. So what is the first question is Mendel's law of independent assortment is best demonstrated in a cross between. Okay, so here they are asking so where the Mendel's law of independent assortment is demonstrated very well means here. So in this, from this you have to answer in which hybrids obviously the law of independent assortment was shown. Okay, so we know that what is the law of independent assortment, the arrangement of the alleles on their individually means without depending on each other or without uh, disturbing each other. Okay, they'll each allele is having its own purpose. So like that, if you have for example see, normally it comes in like taking 2 or 3 characters, so that the for example the height and weight does not belong to each other, right? So height is separate and weight is separate. Like that only here the Mendel's of independent assortment also, it obviously occurs in 2 different traits and which are not dependent or which will not like disturb each other. So normally obviously what were the options given here are first one, 2 monohybrids. They are saying that it occurs in between the 2 monohybrids. No, why? Because obviously you should say that monohybrids will have the similar characters, so they can clumps and everything. So this is not the right answer. If we move on to the next one monohybrid and one dihybrid also, this is also wrong. Why? Because both of them should have the same thing, like they should have their crossing or everything hybridity is same. So this is also not. And third one is 2 dihybrids. Obviously you can consider it as a correct option. We'll move on to the next one so that we can confirm it. Okay, so what was the last one homozygous dominant and one recessive means here they are asking one dominant and one recessive. Obviously we know that if there is a dominant character means it will depress the action of the recessive. So this is also wrong answer. So what is the right answer here is option number C. Okay, so it occurs normally, law of independent assortment occurs in between 2 monohybrids. Okay, so we'll move on to the next question. What is the next question is what would be the genotypic ratio of the monohybrid cross? So this question was came from the monohybrid cross itself. Oh, so, okay, we'll see the options. What were the options given here are they have given the 4 options here. In this we have to say what is the genotypic ratio of monohybrid cross. If we see what is a monohybrid cross, here you can say that normally what the Mendel was done means he have when he had doubt that how the traits are coming and everything, he crossed the 2 different plants which were different in their traits. So one tall plant and one dwarf plant. In the first generation he got the tall plant with two traits from each the parent. Okay, so and 2 alleles from each the parent. So and obviously when he saw all the tall plants, he had doubt that why when we crossed the dwarf plants, why we got the tall. So what he did was he again crossed those tall plants, like the F1 generation tall plants with each other means the result was like amazing. What happened was when he saw that obviously when the plants are appearing tall like physically, but when they crossed with each other the result was like some of the plants are small. So that then he understood that okay, the appearance of the any organism will not depend on the physical characters, it also depends genotypically. So means inside the genes are also responsible for all these things. So that's why like this he crossed the plants, and what was the result he got was like some of them are monohybrids, like homozygous means which have the same alleles or same genes. Some of them are heterozygous, and some of them are like again homozygous. In that also homozygous, some of them are like homozygous tall, heterozygous tall, and then homozygous dwarf. Like that the results were came. So this is by this, obviously physically phenotypic ratio means physically we can see that there are 3 plants here. That's why 3:1 ratio. Whereas if we see it genotypically, means the genes that are present in Y, because this is having equal genes, so this is 1:2, and these two are having 2 different alleles, means one tall and one dwarf, one tall and one dwarf. That's why 2:2 and two dwarf genes. So 1. So 1:2:1 is the genotypic ratio of the monohybrid cross of Mendel. Okay. So that is the result he got from the crossing itself. So here when you see the options here, obviously you can confuse. Why? Because they have given the 2 ratios here which were present in the monohybrid cross. So what was the best option is 1:2:1. Obviously I said this is the correct one. Okay. So 3:1 is also correct, but it's not a genotypic ratio, it is phenotypically. And 2:1 is wrong. And 9:3:3:1 is not happened in the monohybrid cross. So what is the correct answer here? Option A. Okay, so that's it. We'll move on to the next question from the genetics. Okay, what is the next question? So this question came from the dihybrid cross itself. Obviously now we learned about the monohybrid cross. So what is a dihybrid cross? Here also they are asking. Here they are asking about the phenotypic ratio. In before they learned about the genotypical, asked about the genotype, and now they are asking about phenotypic ratio. So that if we know what is a dihybrid cross, you can say it easily. Okay. So what is a phenotypic ratio means physical appearance, right? So physical appearance of the plants. So in the dihybrid cross, we know that the Mendel took the 2 characters of the plant, like he took the green piece and yellow piece, which were in that what they'll say that yellow and round green piece like piece were crossed with the wrinkled and green piece. Okay, so he crossed these two to see that when we cross the characters, like 2 different characters and 2 different colors, means which one is having 2 different characters, what will happen. So in the dihybrid cross, in the first generation, obviously we know that in the monohybrid, he got all of them like appeared like round and yellow. Okay, for the physical appearance they appeared like round and yellow. So again when he crossed that F1 generation piece with each other, what was the surprise is he got some of them are with round yellow, means homozygous round yellow, and some of them are homozygous and heterozygous, both of them, and some of them are round green, some of them are wrinkled yellow, and some of them are wrinkled green. Why? Because when he crossed the heterozygous gametes with each other, obviously he got so many different combinations of the egg cells, like the results. Okay, so like this he got, but when he got so many like round yellow ones, like in number, although they have different types of gametes like homozygous and heterozygous, but they appear round and yellow, right? So that's why here what they are asking about phenotypic ratio. So phenotypic ratio, if you see that, what is the phenotypical ratio of F2 generation? 9:2 means 9 round yellow, 3 round green, 3 wrinkled yellow, and 1 wrinkled green. So this is the phenotypical ratio that was came from the dihybrid cross. Okay, if we see the like what they'll say, genotypical ratio means, obviously you can say that I have to tell you one thing that my sir used to tell me, like in my intermediate, to remember what is that is 1:2:1, 1:1:4, like that, 1:2:1, like that. This is the genotypical ratio of what is that? Yeah, this one. Dihybrid cross. Okay, so here in the question itself we see that they are asking about phenotypical ratio, no? So what is the right answer here? 1:2:1, first option, it's not right, whereas the second one is 3:1. These two belongs to monohybrid cross, so these are not right. And third one is 9:3:3:1, and obviously we know that this is the right one. And fourth one is obviously wrong. 1:1 will not come in the dihybrid cross. Okay, so here the option C is correct answer. Okay, we'll move on to the next question here from the genetics. So what is the next question? Here the question was came from the blood groups. Okay, so blood group O. Here what they are asking in this question is the kid got the blood group O. Consider that the father is having a blood group A and mother is having the blood group B. What will be the possible genotypes they are asking? Means the father and mother are having like A and B, groups here, like father is having A and mother is having B, but the kid got the O. So here they are asking what are the genotypes that were responsible that the kid got the O blood group. So you have to see, obviously one thing you have to remember, that if the parents are having the homozygosity or the equal number of alleles, means dominant alleles in each of them, obviously the kid will get either A or B, sorry, A or B. Normally if he is having the homozygosity, means like A, B means he'll get A or B or AB groups, right? So normally you will consider these groups, but if the kid got the O blood group, means you have to think that how he got, means either one of the parents also, if either one of the parent is having the recessive gene also, he will not get the O. Why? Because for the O blood group to come, obviously both the parents should have the recessive alleles equally. Okay, so that's why you, according to that, you have to consider the option here. Let's see the options. So what was the option? IA and IA and IB. For this option, the options, like the chances were either A or B. No, AB only. Okay, when we cross each other, they'll get the AB only from this combination. Okay, if we move on to this one, like IA and IBI, there are chances that he can get A or B or AB or O also. Why? Because when these two alleles will combine, we'll get the O. Okay, for this combination of genotypes, you can get 4 blood groups, means AB, AB and O. Okay, so if we see the next one, what is that? They have given IA and IBI. For this blood group, you can get AB or A. That's it. Okay, no B. Why? Because it is not having the B here. It can get either AB or A. Okay, if we move on to the next option, from this also you can get that either AB or B for this. Why? Because both of them are not having the A here. So these are the chances. So in these 4 options, what was the best option that the kid can get O group is nothing but IAI and IBI. So from this, when these two recessive alleles will cross, obviously he'll get the O group here. So this is the correct answer. Okay, so what is the answer here? B. B is the right answer. Okay, this is the question from the blood groups. We'll move on to the next one. So what is the next one? Here they are asking about the base sequence of one strand of DNA. Okay, so the base sequence of the one strand of DNA is, here they are asking about the DNA base sequence. Okay, so if you see that, we know what is a base sequence, like A, T, G, G, C, A. If you know that DNA and nitrogen bases, obviously you know that A always combines with the T, whereas T always combines with the A. 1 minute. And the G always combines with the C, right? And C always combines with G, and A always combines with T, right? Means adenine bonds with the thymine, and thymine obviously bonds with the adenine, whereas guanine bonds with the cytosine, and cytosine bonds with the guanine. So this is the basic rule of the nitrogen base pairs. So if we see the options here, you can consider what will be the sequence of this genetic code is. If one strand is having this one, so obviously what is the opposite strand will have, first option they have given is ATGGCA. You see that? If here we are having the A, means here the A will not come. So this is the wrong one. And next one, if you see that TACCGT, you see TACCGT. So the second one is the right answer here. So no need to go to the other options. If you want, you can see that. Third option is TACCGA they have given. In the last, this is also wrong. And first one G they have given. This is also wrong. Why? Because A always pairs with the T here. So what is the correct answer? Option B. Okay, so like this, if you remember which one combines with the which one, you can get the answer easily. And another thing you have to remember is the bonds between the adenine thymine are always double bonds, and between the guanine and cytosine, or cytosine and guanine, are all triple bonds. Okay, we'll move on to the next question. So what is the next question here is, the genetic code is called degenerative. So this also came from the amino acids and genetic code. They are asking why the genetic code is known as degenerative. Degenerate means it cannot regenerate. Why? Because if they have given the so many options here, in that the first one is it has stop codons. They are degenerative because of the stop codons. No. Why? Because stop codons are for stopping the transcription or translation, right? So this is also, this is not right, sorry, translation. So this is wrong. And excuse me. And the next option is one codon codes for multiple amino acids. You can see this option, one codon will not code for multiple amino acids. If it codes like this, obviously you will not get a best sequence, so that we cannot get different types of DNA, RNAs, so like proteins. So this is also wrong. If you move on to the third question, multiple codons code for a single amino acid. This is right. Normally this option is right. Why? Because a single amino acid can contain so many codons. Okay, this is right. And if you move on to the fourth option, it has overlapping triplets. The triplets will not overlap, but overlapping triplets will not cause the degeneration. Okay, so what is the correct option here is, because the genetic code is degenerative because it is having that multiple codons code for a single amino acid. That's why it cannot be regenerated. Okay, this is the correct option here. Okay, so we'll move on to the next question. What is the next question is, which enzyme is responsible for unwinding of DNA during replication? So we'll move on, moving on step by step, like DNA and RNA, like amino acids, and then from the process replication. So replication means we know that the breaking of DNA to form the kits, like to form the next generation DNA, right? So like that, if you see that what is the replication and how it will takes place in this image, if you see from the question, here they are asking what is which enzyme is responsible for unwinding or breakage of the DNA, the double stranded DNA. Okay, so here they have given the so many options here. First one is DNA polymerase. So if we see the functions, DNA polymerase, obviously like what it will do, it will produce more amounts of DNA molecules, okay, by adding the nitrogen bases, like polymerization, by doing the polymerization. Okay, next one is ligase. Ligase is the enzyme which is responsible for the attachment of the strands. Okay, the broken strands were attached with the help of ligase enzyme. So this is the function of ligase. Whereas helicase means this is a or they'll say this is also one enzyme which is normally responsible for breaking the DNA, means mother DNA, into two strands. Okay, so primase means this is the enzyme which starts the replication process. Okay, so in these four options, by seeing the question, you can say that unwinding means nothing but breakage of DNA. So what is the correct answer here? This one means helicase is the enzyme which is responsible for the breakage or unwinding of the DNA in the replication. Okay, if we move on to the next question that was given in NEET 2020 from the replication itself. So what is that? Which enzyme is required continuously during replication of lagging strand? We know that in the DNA replication we are having leading and lagging strands, right? From the leading strand itself you can get the continuous DNA, whereas in the lagging strands you'll get some of the pieces. Okay, Okazaki fragments are formed. So which enzyme is required for continuously during replication of lagging strand? Why? Because without this enzyme the lagging strands cannot replicate. Okay, cannot show replication. So what is that enzyme? Is lagging strand continuous replication, if it have to happen, means the Okazaki fragments have to combine with each other. So for combining the Okazaki fragments, what is the enzyme that plays major role is ligase, right? So ligase is the enzyme which is responsible for the continuous replication of lagging strand here. So here they have given the options. First one is the right one, obviously, and another one is DNA polymerase, RNA polymerase, and helicase. So these are all wrong. That's why option 1 is the right. So these are the questions that came from the replication process. Okay, so so still now we learnt some of the questions from the genetics chapter, as there are some other also. So we'll see it in the next video, because it will become very lengthy. That's why I'm stopping here. So you can expect some of the questions from the part 2 itself. Those are like this I have given here, and there are some other also. If you want to watch the video, just try to like try to watch the next video itself, so that you can get the answers of these questions and the other ones also. Okay, thank you guys, thank you so much. I hope all of you understood these PYQs and FAQs from the genetics chapter. We'll move on with this next video also. Just try to catch up with the other videos also. Thank you. And if you like the videos, just share it with your friends. And if you want to see my videos and everything, the information I'll give you, just subscribe to my channel and stay on with me. Okay, thank you.